Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar, titled Improving Cell Therapy Manufacturing with Online Monitoring of Critical Process Parameters During Cell Expansion. I'm Casey Nevins, an editor at BioInsights, and joining me today are Boris Aleynik and Mindy Miller, who will explore how efficient monitor and control of glucose and lactase levels in the culture environment can influence T-cell differentiation. After the presentation, we'll have a live Q&A session. We invite our audience to pose their questions to Boris and Mindy using the Ask a Question box at the bottom of your screen, and we'll try to get to it during the session. Now I'd like to introduce our presenters. Boris Aleynik, Director of Strategy and Product Marketing at 908 Devices, leads marketing strategies for life science products and has prior experience as Director of Strategic Product Planning at PAL Life Sciences. Mindy Miller, a senior scientist and manager at Terumo Blood and Cell Technologies, leads a team developing innovative cell therapy solutions and has a doctorate in immunology, postdoctoral experience, multiple publications, and a Young Investigator Award from the American Association of Immunologists. So, without further ado, I'll hand you over to Boris to kick us off with the first presentation.
Thank you for the introduction. Welcome, everyone. Thank you for joining us. So first, let me tell you a little bit about our company. So 908 Devices was founded in 2012 in Boston, Massachusetts, where we still manufacture our products. Our mission is to make chemical and biochemical analysis simple, smart, and speedy. We focus on impactful applications in forensics and life sciences, where quick and robust answers are needed, sometimes in life or death situations. We have a broad range of customers across biopharma, government, and academic institutions. Our devices are installed in all the top 20 biopharma companies. We have an award-winning portfolio of process analytical technology solutions used in development and manufacturing of biopharmaceuticals. Our devices are used to measure and control critical process parameters and critical product quality attributes. In 2016, we launched ZipChip, a capillary zone electrophoresis sample separation platform for mass spectrometry, used for characterizing biotherapeutic proteins and analysis of small molecules. In 2019, we released the Rebel desktop device, what process development scientists can use in their lab to analyze concentrations of over 30 media components, including amino acids, biogenic amines, and vitamins, to screen and optimize media and feeds. Rebel provides results in about 10 minutes and is very easy to use, so you don't have to send your samples out to a lab and wait days or weeks for the results. And last year, we launched MAVEN and Maverick for real-time monitoring and control of bioprocess parameters. MAVEN uses enzymatic biosensing technology for glucose and lactate measurements, and Maverick is a Raman spectroscopy-based analyzer for glucose, lactate, and biomass. Unlike conventional Raman analyzers, Maverick does not require costly and complex empirical calibration. No chemometric modeling is required. Just perform a simple 2-point calibration of the probe, and you are ready to monitor and control your bioprocess. So let's talk about cell and gene therapies. Cell and gene therapies have emerged as promising treatment options for previously incurable diseases. As of this year, over 2,800 developers globally are working on CGTs, with over 1,700 ongoing clinical trials, about half of which are non-industry sponsored. So the CGT market is projected to grow to over $80 billion by 2029, with significant upside from non-oncology indications. And despite the sustained investment in rapidly growing pipeline, the high cost of manufacturing, high cost of treatment, and limited availability still pose major challenges to the mainstream adoption of these treatment modalities. Reducing wait times and improving manufacturing consistency are key to making these therapies more affordable and accessible for patients, and hopefully help establish them as first-line treatments. So why are cell therapies so difficult to manufacture? Cell therapy manufacturing processes are complex, difficult to characterize, and highly labor-intensive. Unlike biotherapeutic proteins, cell therapies require retention of cell function and quality. Donor-to-donor or patient-to-patient variability of starting material can lead to very different outcomes for the final product. Recent FDA guidance emphasizes well-controlled manufacturing processes. This can be achieved via in-process control and in-process testing at relevant time points. In-process testing should evaluate multiple parameters such as viability, cell count, phenotype, and others. And the results can be used to guide manufacturing decisions, for example, when to exchange the media and when the cells are ready to be harvested. So it's quite clear what cell therapy manufacturing would greatly benefit from robust automation and integrated process analytical technologies. Cell expanded in vitro, influenced by various factors like cytokines, patient disease status, and culture medium composition. Additionally, small changes in culture conditions can significantly affect cell metabolism and differentiation. Automated systems with real-time monitoring and control can achieve higher consistency in yields. However, historically, automated systems had limited real-time monitoring capabilities, instead relying on manual sampling and analysis of critical parameters by their operators. This is labor-intensive, prone to human error, provides limited information about the state of the process, and increases risk of contamination. Careful monitoring and control of critical parameters with minimal human intervention is crucial for process robustness and is best achieved with the help of real-time monitoring technologies integrated with the automated manufacturing systems. There are multiple parameters that are important to monitor and control during cell expansion, but there are two in particular that we'll be discussing in more detail today, the glucose and lactate. Glucose is essential for cell growth, and its concentration has been shown to impact certain important attributes, such as differentiation and phenotype, as well as having an impact on the efficacy of CAR-T treatment after the cells have been infused back into the patient. Excessive lactate can indicate suboptimal culture conditions and inhibit cell growth. Optimizing glucose levels while maintaining lactate are crucial for robust cell expansion and final quality of cell products. That is why we developed MAVEN. MAVEN offers continuous monitoring of glucose and lactate with up to 30 measurements per hour. It provides precise monitoring even at low concentration levels without loss of bioreactor volume while maintaining process sterility. Multiple data communication options and integrated PID controller make integration with process control and data acquisition systems straightforward. We designed it with ease of use in mind to help operators focus on value-added tasks instead of spending all this time on manually taking and analyzing samples, especially during off hours when access to the lab may be limited and when reducing clean room dwelling time is desirable. The MAVEN system includes a single-use bags for clean and spent transport buffer fluid, internal collaboration solution, enzymatic biosensor, a gamma-sterilized flow cell that welds into the Quantum's Flex cell expansion system's consumable, and a tubing set connecting everything together. The device conveniently mounts on Quantum Flex's back pole, as you can see in the image on the right, and the real-time data can be viewed via web browser on a PC connected to the local network. So let's take a closer look at how MAVEN works. At the beginning of the measurement cycle, clean buffer solution is pumped from the buffer bag into the flow cell, and the buffer flow is paused. The flow cell has a semi-permeable membrane that separates the buffer from the process environment inside the bioreactor. The cutoff rating of the membrane limits the molecular weight of things that can pass through it. Glucose and lactate molecules are quite small, so they can diffuse into the buffer solution, but large molecules, cells, and also any kind of bacteria or virus are too big to pass through the membrane. Hence, there is no bulk sampling and no volume loss. Once glucose and lactate had time to load into the buffer flow is resumed, and the loaded buffer is sent to the sensor. The concentration of glucose and lactate is determined by the highly selective enzymatic biosensor, which generates an electrical signal proportional to the concentration of analytes in the process. Lastly, the buffer is disposed of into the buffer waste bag, and the cycle repeats as often as every 2 minutes. Integrating MAVEN with Quantum Flex is quite straightforward. You would remove the MAVEN flow cell from the sterile pouch, mark welding locations on the EC tubing of the Quantum Flex consumable, and use the sterile tubing welder to weld the flow cell in. After, you would follow the rest of the setup protocols for both devices. With that, I'll pass it over to Mindy so she can tell you more about the QuantumFlex cell expansion system and show what the online glucose and lactate data would look like.
Great. Thank you, Boris. Great description of the 908 MAVEN. So I'm here today from Terumo BCT to talk about how the 908 MAVEN device can be integrated into Terumo BCT's QuantumFlex cell expansion system for automated glucose and lactate sampling. Before I do that, I'd like to quickly provide an introduction to the QuantumFlex platform for anyone who isn't familiar yet. It's a benchtop device that can be used to expand both adherent and suspension cells in a functionally closed and automated manner. Inside of this device, the core expansion technology comprises a hollow fiber bioreactor. You can choose either the larger standard size or the 1/10 small scale version. As part of the platform, there is also a software application called the Cell Processing Application, or CPA, which allows a user to program each step of the cell expansion process remotely from their desk. My team has found this feature to be tremendously helpful because they don't need to gown up and go into the laboratory just to program the device. It definitely saves a lot of time for the operator. CPA also allows you to manage an entire fleet of devices and does so in a manner that facilitates 21 CFR Part 11 compliance. So let's take a deeper look at the core technology of QuantumFlex. Inside the unit contains a disposable kit with the hollow fiber bioreactor in the center, where the cells are maintained and expanded, a sampling port for taking manual samples of the culture media, and a gas transfer module that oxygenates the culture media. Cell culture media is brought into and out of the system through the inlet and outlet lines, and fluid is directed by various pumps. All of these provide the user with a highly customizable options for ensuring their cells experience the perfect microenvironment for expansion. The real beauty of this system is within the bioreactor itself. A simple way of describing the system is to imagine a hollow tube filled with a bunch of tiny straws. For the standard bioreactor, there's actually 11,500 of these tiny straws, which we call hollow fibers. And each of those fibers are semi-permeable, meaning that cells are kept inside of the fiber, but media and gases can pass through. This ultimately creates two different spaces within the bioreactor. There's the intracapillary space, or the IC space, where the cells reside, and the extracapillary, or EC space, outside of the fibers, where fresh media and oxygen is perfused. A great feature of the system is that the semi-permeable nature of the hollow fibers actually retains larger proteins like cytokines and concentrates them with the cells, meaning that you can use a lot less of the most expensive reagents. And another one of my favorite features of this system is that it's physiologically similar to how our bodies operate. So if you think of the human body, cells are usually no more than 200 microns from a capillary or a source of oxygenated blood and nutrients. And in this system, our hollow fibers are 200 microns in diameter, meaning that our cells are kept in a very physiological environment. Here are some examples of cells and applications performed on the quantum system. Adherent cells, such as iPSCs, mesenchymal stem cells, fibroblasts, endothelial cells, a whole variety of adherent cells have been expanded on hollow fiber, even suspension cells. And we're able to grow suspension cells by changing the direction of the fluid. So basically, we push fluid in from both sides of the bioreactor to maintain the cells within the fibers. And cells like T cells, Tregs, CD34s, even NK cells have been grown on this platform. Now, because we can do such interesting fluidic manipulations, we can also perform different applications, such as vector production using adherent cells, exosome production, and even gene modification. So let's get to the data. This is an example of a CD3 positive T cell expansion. In this experiment, we expanded 30 million T cells on the standard bioreactor and 6 million T cells on the small bioreactor. The cells were activated inside the bioreactor with Immunoccult soluble T-cell activator and cultured with ex vivo 15 media. These cells were grown for 9 days under perfusion feeding with media recycling starting at day 5. At harvest, the standard bioreactor yielded over 30 billion T cells, and the 1/10 scale small bioreactor was over 3 billion T cells. So to describe how the T cell expansion kinetics look on the device, on the left-hand side, you can see the standard bioreactor growth curve. As per typical T cells, there is a lag phase for several days, followed by this log expansion. Whereas by day 8, we have well over 2 billion cells. In the small bioreactor, we similarly see a lag phase followed by the log phase expansion. And you can see that we hit the 2 billion cell mark just after day 8. That is compared to a flask expansion, whereby we didn't quite achieve a full 2 billion within that 8-day mark, but you can see a similar T cell expansion trajectory. So as the cells grow, feed rates are increased to accommodate metabolic requirements. How do we know when to increase rates? So by using lactate and glucose, we can actually determine when to change the feed rates on the device. So you can see that lactate grows over time in the culture and is maintained below the threshold of 13 millimoles per liter, which we just set for this run. We really like to measure lactate because it tells us exactly when to increase the feed rates and can actually save on media consumption so that we don't increase feed rates prematurely. On the left-hand side, you can see lactate from the EC sample port on the standard bioreactor. And just after day 6, the lactate levels were approaching the threshold that we did not want to exceed. Therefore, we increased our feed rates to bring the lactate levels back down as lactate was washed away into the EC side of the system. In the small bioreactor, lactate levels were naturally kept a bit lower, so we didn't have to aggressively feed as much. And that's quite different from what you see in a flask sample, where each time you do a media exchange, you have wild fluctuations in lactate levels. Likewise, measuring glucose can also indicate when a run may need increased feed rates. You can see in the standard bioreactor, over time, the cells were consuming more and more glucose as the cells themselves were expanding. In the small bioreactor, you can see that early dip in glucose, meaning that we actually did increase feed rates to ensure that our cells had enough glucose for the rest of the culture. And again, in the flask sample, you can see the different glucose levels during media exchanges. So now imagine that you're expanding adherent cells. Now, classically, one would look at these cells under a microscope and estimate confluence. However, in a bioreactor, it's not so easy to visualize the cells. We have found that we're able to calculate a lactate production rate, and that this rate correlates with cell expansion. This can give the operator insight into how the cells are expanding and even indicate when to harvest. And we've used this method for both MSCs as well as iPSCs. And this data here shows specifically what the lactate generation rate looks like for iPSCs. Since measuring glucose and lactate is so important in the quantum system, we were excited to try it with the 908 MAVEN device to have a fully automated sampling process. We tested this with a JERCAT cell expansion and were able to sterile weld the flow cell into the EC loop tubing prior to loading the set onto the device. This connection procedure is quick and easy to perform with a Terumo BCT welder at the benchtop. The automated sampling was a great feature. To be able to set it up and just automatically take samples is great from the usability perspective, but we were actually surprised with how good the resolution was. By taking samples more frequently, you're able to really pick up small changes in the culture. You can see on both of these graphs, but is especially prominent in the lower glucose graph, each time we change the feed rate. This can be really helpful for maintaining cells that are sensitive to culture changes, and it may advise to use perhaps performing feed rate changes more frequently than once per day to decrease that spike. Likewise, if I were using lactate generation rates to understand when to harvest, having real-time data may indicate an earlier harvest rather than waiting until the end of the day lactate measurement. So overall, the ability to reduce the hands-on time as well as detecting these very, very fine changes, was quite impressive. So in conclusion, glucose consumption and lactate generation are key metrics in monitoring a quantum flex cell expansion. These lactate production rates correlate with adherent cell yield and can be used to monitor the health of expansion and harvest date. Using the MAVEN device allows for real-time automated glucose and lactate sampling and enhances the detection resolution to maintain tight control over culture conditions. Automated systems like this ensure reproducibility and reduce the amount of hands-on time required from operators. If you'd like more information on either the 908 device's MAVEN or the Terumo Blood and Cell Technologies Quantum Flex Cell Expansion System, please scan these barcodes below. We're happy to take any questions, and I will hand it back to the moderator.
Thank you so much, Boris and Mindy, for a great presentation. We're now going to start our Q&A session, so I'd like to invite you to join me on camera. Our first question is for Mindy. It says, in quantum, since the IC and EC compartments are separate, is it possible to utilize different media types in each?
Yeah, I love that question. So absolutely. We actually hang separate media bags. So we hang a media bag for the IC side as well as a media bag for the EC side. So for example, if you're growing T cells, your IC media would contain perhaps serum, probably IL-2, whatever other cytokines you want to include, where the EC side is just going to be base media. And that really helps cost saving when it comes to these large cultures.
Thank you so much. Our next question is for Boris. It says, how much sample is consumed during the measurement?
Sure. So MAVEN utilizes a semi-permeable diffusion membrane, and it has molecular weight cutoff to only allow the small glucose and lactate molecules to diffuse across it into the transport buffer. So there is really no bulk sampling of the media and no loss of the actual volume or the material from the bioreactor.
Thanks for that, Boris. Back to you, Mindy, for our next question. It is, how would you modify a quantum run if it was critical to keep lactate levels very low?
Yeah, so sometimes people will want to culture their T cells and they're really careful to look at lactate levels. Like maybe they only want to keep lactate below 8 or 10 millimoles per liter. If that's the case, the best way to do that is actually to increase the EC circulation rate. So if you increase the EC circulation and the EC feeding, what that's going to do is pull in more base media and allow that lactate to diffuse across the membrane and be washed away into the waste container, for example. And that really helps control lactate tightly. So if you do want to take very frequent lactate readings with the MAVEN, you can get super high resolution of that data and then modify your run accordingly to keep that lactate level really low by increasing EC feed.
That's a great answer, Mindy. Thank you so much. Our next question can be open to either of you. It says, do you have to generate new lactate production or cell density curves for each different cell line?
Yeah, so that's a great point. Yes, the short answer is yes. So each different cell type is going to have a different baseline for lactate generation. Some cells are super high lactate producers, some aren't. Some cells can even utilize lactate. So it definitely is something that you would want to generate a new curve for each particular cell type.
Thank you, Mindy. Our next question is, could you please superimpose the feed rates with the lactate concentration changes?
Yes, that would be a great way to look at the data. The way that we presented it on that graph, basically we highlighted when those feed rate changes, but certainly that would be a great way to look at the data.
Thanks for taking those, Mindy. Our next question goes back to Boris. It is, how long does the biosensor last?
Yes, the biosensor is good for about 5,000 measurements or 30 days.
Thank you. Mindy, can the disposable set be reused?
So these are single-use disposables. So the disposable kit actually comes on, so how do I describe this? Let's see. It's a kind of a hard plastic surface that you mount into the bioreactor, and that disposable kit contains the bioreactor itself, all the tubing lines. And so basically it's loaded on prior to the cells. The cells are, put into the set, they're expanded, they're harvested, and then that disposable set is removed and discarded. So each particular expansion would require a new disposable kit.
Great, thank you. Our next question is for Boris. It is, oh, sorry, I started reading the wrong one. Our next question says, could you please explain why adherent T cells lactate production rate correlates with yield? Then they say, you mean reducing lactate will help improving yield? How to reduce lactate levels? By feeding more media?
Okay, so I did present a lot of data in pretty short amount of time. So we are not showing data for adherent T cells. T cells are suspension by nature. And so for T cells, when we're growing them, we really are only concerned with keeping the lactate levels within a certain limit. And so in order to do that, we could increase the EC feed and EC circulation rate, and that would help keep the lactate low. When I showed the data talking about the lactate production rate and how that correlates with yield, that is very specific to adherent cells. So the adherent cell data I showed was from MSCs and iPSCs. So these are the cells that we can't necessarily pull a sample of the cell themselves to measure cell density, for example, because they are adherent to the bioreactor. So to determine how many cells we have in the culture and at what point we would want to harvest those cells, that is when we really use that lactate production rate to help correlate.
Thanks for clarifying that, Mindy. Now we're going back to you, Boris, for our next question. It is, what is the shelf life of MAVEN consumables?
Yes, it's 1 year for the biosensor and the calibration standard, 2 years for the flow cell, the tubing set, and the buffer bags, and for the buffer powder packs, it's 3 years.
Thank you so much. Mindy, is it possible to grow cells in hypoxic conditions in quantum?
Yeah, this is becoming a super hot topic, especially as we start to understand more about stem cell memories, growing TILs, for example. Anytime that you know your cell prefers a microenvironment that has less oxygen. For the quantum system, we actually have a gas inlet port. So we can port in any gas mix that would be appropriate for your cells. So if you do want to try to grow cells in a hypoxic condition, you could either pipe in directly what particular gas you want, or we can also connect a gas mixer. So if you have independent tanks for your different gases, you can use a gas mixer to change that. And what's cool about that is throughout a run, you could even modify the amount of oxygen going into your culture. So say you want to start the run at normal oxygen concentrations, and then throughout the run, change it to be more hypoxic. That's also possible using a gas mixer.
Thank you, Mindy. Our next question goes back to you, Boris. It is, how much buffer fluid is needed for a typical run?
Yeah, so the buffer comes in powder form in packets, and each packet is good to prepare about 5 liters of the buffer fluid. And the frequency of measurement and obviously the duration of the run will determine how much buffer is used or consumed. So for example, at about 30-minute frequency interval, right, and measurement frequency, you would consume about 0.4 liters per day of buffer. And then you would either replace it if your run is longer, or but typically it would last long enough for one run.
Thank you so much. Again with you, Boris. What data communication options are available?
Sure. There are multiple communication options which MAVEN supports. It includes serial, analog, Modbus, TCP, OPC UA, and also you are able to export the run data in a CSV format for later analysis.
Well, thank you so much, Boris and Mindy, for answering these questions.
You.
Unfortunately, that is all we have time for today. Any questions we didn't get to, we will reply to by email. The webinar will be available on demand tomorrow, so look out for an email from us with the link. All that's left is to thank Boris and Mindy once more for a great presentation, and thank you to the audience for listening. We hope you'll join us again soon.
Thank you.
Thank you.